Hi everyone, my name is Ajay Reddy, and the molecule I'm presenting today is dexamethasone. So to begin with a quick overview of this presentation, I'm going to cover structure and drug-like properties, then the discovery of dexamethasone, followed by its therapeutic uses and side effects, followed by a mechanism of action, pharmacokinetics, and competitor drugs. So, dexamethasone is a synthetic glucocorticoid medication. Glucocorticoids are a class of steroid hormones. You can see the structure of dexamethasone here. Note that it has the characteristic 4 rings of a steroid. It also has 3 hydroxy groups, 2 ketone groups, and a fluorine atom substituent. And finally, there are 8 chiral centers in dexamethasone. Moving on to the drug-like properties. So dexamethasone does meet all of the criteria in Lipinski's rule of 5s. It has 3 hydrogen bond donors, the 3 hydroxyl groups, which is less than the cutoff of 5 hydrogen bond donors. It also has 5 hydrogen bond acceptors, which is less than the cutoff of 10. And all the hydrogen bond acceptors are oxygen atoms. Dexamethasone has a molecular mass of 392.5 daltons, which is less than the cutoff of 500. And it also has a log P of 1.83, which is less than the cutoff of 5. Moving beyond Lipinski's rules, Weber's rules characterize additional important drug-like properties. So note that dexamethasone has 2 rotatable bonds, which is less than the recommended cutoff of 10, and the polar surface area is just under 100 angstroms squared, and that's well under the recommended cutoff of 140 angstroms squared. Moving on to how dexamethasone was discovered. So Philip Hench and Edward Kendall first discovered cortisone and isolated it from the bovine adrenal cortex. In 1948, Hench successfully used cortisone to treat a patient with severe rheumatoid arthritis. It was soon discovered after that cortisone was a prodrug of the compound cortisol. Cortisol is a key anti-inflammatory compound in the human body. Cortisol found uses as a treatment for Addison's disease. So cortisone and cortisol are classified as corticosteroids. There are 2 main categories of corticosteroids: glucocorticoids and mineralocorticoids. Glucocorticoids tend to have positive effects on human health, while mineralocorticoids can be harmful. While cortisone and cortisol have many beneficial pharmacological properties, they also cause unwanted side effects. These compounds possess mineralocorticoid activity, that is, they lead to water and salt retention. They also cause metabolic side effects like weight gain and increased blood sugar levels. In 1954, Fried and Sabo at Squibb prepared a series of analogs of cortisone. They found that 9-alpha-fluorohydrocortisone, or fludrocortisone, was 10-fold more potent than cortisone in relieving rheumatoid arthritis. Unfortunately, the compound's off-target mineralocorticoid activity was increased by 300 to 800-fold, which caused water and salt retention. You can see the primary difference in fludrocortisone is they change that hydrogen atom to a fluorine atom. Next, cortisone was fermented in the mold broth, Corynebacterium simplex, and underwent microbial dehydrogenation. This led to the isolation of the novel steroid prednisone and prednisone's active metabolite prednisolone. Both prednisone and prednisolone were approximately 5 times more active than cortisol and possessed less off-target activity. Specifically, they produced less salt retention. So a boy taking cortisone submitted a urine sample to Liderle Laboratories, and from this they were able to isolate a corticosteroid with an added hydroxyl group. This likely resulted from cytochrome P450 metabolic oxidation. The hydroxyl substituted analog was merged with fludrocortisone to produce triamcinolone. This drug was as potent as prednisolone, but possessed far less off-target mineralocorticoid activity. You can see for triamcinolone, the hydroxyl group that I just mentioned is at position 16, and you can also see the fluorine atom. So then Merck chemists synthesized analogs of triamcinolone with a methyl group in place of the hydroxy in an effort to inhibit metabolic reduction of the side chain's ketone. The methyl group turned out to be easily metabolized by cytochrome P450 and consequently did not prevent reduction of the ketone. However, the resulting compound proved to significantly depress mineralocorticoid activity while enhancing the anti-inflammatory activity by 6-fold compared to prednisone. Dexamethasone also possessed an extended duration of action compared to hydrocortisone. So that's how dexamethasone was discovered. We can now turn to what it is used for. Dexamethasone is primarily used as an anti-inflammatory agent. You can see its effects on immune cells in the top image. Note that it inhibits the functioning of multiple types of immune cells, including B cells, helper T cells, neutrophils, basophils, and eosinophils. Dexamethasone can thus be used to treat a number of inflammatory and autoimmune disorders, such as rheumatoid arthritis and bronchospasm. Other inflammatory conditions it is used for include laryngotracheobronchitis, and that's a type of respiratory infection usually caused by a virus, and plantar fasciitis, which is a disorder of the connective tissue supporting the arch of the foot. It can also be prescribed to decrease swelling after dental surgery. It's present in some eye drops that are prescribed after eye surgery, and dexamethasone also helps to minimize the inflammatory response to an implanted cardiac pacemaker. Next, dexamethasone is used in the treatment of cancer, either directly as a chemotherapeutic agent or to counteract the development of edema in the brain or the nausea induced by chemotherapy. Finally, dexamethasone has shown some promise as a treatment for COVID-19. It is recommended by both the UK's National Health Service and the United States National Institute of Health for COVID-19 patients who require supplemental oxygen or mechanical ventilation. It does help treat acute respiratory distress syndrome. A multi-center randomized controlled trial of dexamethasone used for that purpose showed promising results. Dexamethasone does, however, have a number of side effects. These include cardiovascular side effects of the central nervous system, the skin, endocrine side effects, metabolic side effects, hematological side effects, immune, musculoskeletal side effects, ocular side effects, hypoxia, and if used improperly, it can actually cause acute respiratory distress syndrome. So moving on to dexamethasone's mechanism of action. If you look at the top picture, you can see that dexamethasone binds to glucocorticoid receptors in the cytoplasm. The bound complex then enters the cell nucleus. There it interacts with nuclear DNA molecules that trigger the signaling pathway for the anti-inflammatory outcome. Classically, glucocorticoid binding to glucocorticoid receptors activates or represses gene transcription, and glucocorticoids regulate up to 20% of the genome according to some studies. This kind of accounts for the wide array of side effects that I just mentioned. So note that dexamethasone binds not just glucocorticoid receptors, but also cytoplasmic mineralocorticoid receptors. These two diffuse into the nucleus. This results in the side effects of water and salt retention. Taking a look at the bottom picture, you can see that glucocorticoids like dexamethasone enter the cell and bind to the glucocorticoid receptors that are complexed with a couple proteins. Upon binding, the receptor dissociates from those other proteins, and the receptor ligand complex enters the nucleus. Regions of the nucleus that bind the glucocorticoid receptor complex are called glucocorticoid response elements, or GREs. Glucocorticoid receptor complexes may homodimerize and bind to GREs, in which case they increase gene transcription. Alternatively, the complexes may remain monomers and bind to either a GRE or other transcription factors. In this case, they repress gene transcription. Returning to the top picture, you can see that dexamethasone can also interact with various membrane-bound G-protein-coupled receptors, which directly or through glucocorticoid receptors also located in the cell membrane. Either directly or through those glucocorticoid receptors. This leads to secondary messenger cascades. Finally, glucocorticoids like dexamethasone exert a number of effects through non-genomic mechanisms. They may intercalate into membranes, affecting cation transport. Alternatively, the ligand-bound receptor complex may translate into the mitochondria, which results in apoptosis. So turning now to the pharmacokinetics of dexamethasone. Under absorption, it has a max concentration of about 14 nanograms per milliliter. It takes about 2 hours to reach that concentration. The area under the curve is about 330 nanogram hours per milliliter, and it's about 70 to 78% bioavailable if taken orally. Under distribution, the volume of distribution for this dose is about 51.0 liters, and the percent of protein bound in plasma is about 77%. In terms of metabolism, dexamethasone is 6-hydroxylated by cytochrome P3A4 to 6-alpha and 6-beta hydroxydexamethasone. Dexamethasone is also reversibly metabolized to 11-dehydroxydexamethasone by corticosteroid 11-beta dehydrogenase isozyme 2. And it can also be converted back to dexamethasone by corticosteroid 11-beta dehydrogenase isozyme 1. Under excretion, less than 10% is eliminated in the urine, which is kind of low for a drug of this class. It has a half-life in the body of about 6.6 hours, and the clearance is about 15.6 liters per hour. Turning to competitor drugs on the market, here are some of the alternative drugs that can be used. Cortisol was one of the first drugs of this class to be used, and so it's therefore used here as a baseline for comparison. You can see that prednisone had greater glucocorticoid activity and reduced off-target activity with a longer duration of effect than cortisol. Methylprednisolone improved on this slightly. Fludrocortisone did have increased glucocorticoid activity, but it had also had a great deal of mineralocorticoid activity. Finally, of the drugs presented here, dexamethasone possesses the greatest on-target glucocorticoid activity and minimal off-target mineralocorticoid activity. It also has the longest duration. That's it from me. Here are my references, and thanks for listening.